Alright, let's go. So what I'd like you to find, look at this. So that's what we did at the end of doing plant tissues and looking at all of these models. I think, yeah. So it should be making sense now, doesn't it? Okay? I'm going to leave the screen. Is it okay? Can you see here? Oh no, there, that's better. Alright, so I don't have a model of a root. And you can't, I've looked on the internet, there's none available in South Africa that are good. Alright, so this is like a millimeter at the end of the root, or maybe a few millimeters. Okay, remember root caps? I've colored everything in yellow, epidermis. So there's the root cap, part of the epidermis that protects the root as it grows. Then anything grey here is parenchyma filling up. So all of that is grey, and even there. And then you've got the middle region where the vascular tissue is found. And the vascular tissue is in the form of a star or a cross, it depends. So it could be that shape, there could be a 3-armed star or whatever. It depends on the different species, what the shape of the xylem is in the middle. So there you're going to find xylem vessels and tracheates. Do I have to pull those all out again, those models? Mm-hmm. Okay. And then in between the arms of the star is the phloem. So if I had blue in my hand now, there would be phloem, wherever. Okay. And then this ring. Okay, now that ring isn't shown here. We'll do it later, next week, Monday. What do you call it? The pericycle and the Casparian, the endodermis. Do you remember me using this thing? And I'll show you how water moves through. Mm-hmm. Yes. And then suberin. Okay, so yeah, so it's not all shown there. So this is called a stereogram. It's a 3-dimensional picture.
So now that circle around is the cortex of.
Okay, so cortex is here.
Oh yes.
And then you've got the parenchyma filling up in between xylem and phloem, and separating those two is where the pericycle, that ring of meristematic tissue to make branch roots, and the endodermis, because you've got epidermis on the outside, endo on the inside. But it's not shown here. The endodermis separates where the vascular tissue is to the outside. And it was made up of those cells with that, which we'll do next week, the suberin Casparian strip. It's a very difficult thing to visualize.
So now the pericycle will be first and then the endodermis.
Let me think, no, it's endodermis, then pericycle. Okay, but we'll do that on Monday. Okay. Alright, then we've got, that's like my model, so the root. So anything coloured in yellow is epidermis. And they've shown how it's peeled off. There's some guard cells, and there would be a cuticle covering it. Okay. Then I've used purple, it looks like. What's the purple? I know it's written here, it's fine. What's below the epidermis, the strengthening tissues? Strengthening? Well, in this case, it's actually parenchyma, those cells with the unevenly thickened walls. And then below that, I've coloured some in green. Remember, I explained all this in the video. Okay, so I've coloured some of these cells in green. What do you think they are? Hmm? Yes, parenchyma with chloroplast. And I can see little air spaces here and there. So that's parenchyma with chloroplast. There you can see it there as well. And then here in the middle is with, yeah, no chloroplast. Pith. Remember pith? P-I-T-H. Then you've got the veins or vascular bundles. Alright, so this is what I always, the other class I said, yeah, we're building with Lego. We're taking tissues and we're putting it together, and we've made a plant now. So now we've got all these different Lego blocks making up the different parts of the plant. That's what we're trying to do. Start with cells, look at tissues, how are the different tissues different, where are they found? And that's what we've got here. Alright, so there's the vascular bundle. On the inside, what always faces the inside? Okay, look here. Xylem. In the leaf, it's phloem below. Xylem at the top of a leaf, but in a stem, the xylem faces the middle, because that's where the wood is going to be. And xylem is wood, remember? Or wood is xylem. Okay, so there's xylem, and then the blue here, separating the two. There, bluey green. Okay, there is meristematic tissue, but it's not shown here. This is a very young stem. So there could be a little row of meristematic tissue, which I would colour in yellow. And the blue is phloem. And then the red on the outside, sclerenchyma fibers. Okay, so let's just do this quickly. So, xylem vessels and tracheas here. Then there, the phloem. They haven't drawn meristematic tissue. What was this called again? Sept tube cell? Companion cell. The blue, there. There, going around. Okay, just tell me again. If it's xylem, what is this? Xylem trachea? Xylem vessel. There, you can see some big ones, little tracheas there. Right, this on the outside is? Fibers, sclerenchyma fibers, lots of them. Okay, running down the back part of the vein. Are we happy? Okay, so now let's look at, easier one I think is the leaf. We've done this lots of times. Okay, so cross section through the leaf, part of the leaf. This one we've done. Let's just see if it matches. Okay, so you can see there, cutting across the leaf, there's a branch vein, mid-vein or mid-rib, it's sometimes called. We don't even really need to know. This is like primary school. Leaf apex. Did you know that the lamina is the leaf blade? It's got a big lamina, it's the surface. It's the big surface. Alright, branch veins, leaf margin, mid-rib is the mid-vein, we call it that as well. PTL or leaf stalk, I just left it there. Alright, so now let's see if it matches. Am I holding it the right way around?
No.
You happy? Okay, but the other, there's a little branch vein, there's a little branch vein. Alright, so what's this? Upper epidermis with cuticle. Okay, then palisade parenchyma. These, they've got more chloroplasts, smaller air spaces, because more sunlight reaches there, more photosynthesis. And below here, yeah, you can't really see the big air spaces here, I must say, but you can see it on here. Okay, big air spaces, lots of gaseous exchange and some photosynthesis. Alright, what's this here going around?
So I'm sorry, how does say parenchyma have chloroplasts and bigger air spaces?
Smaller air spaces, you see how squashed they are? Yeah, they're more squashed but spongy, like a sponge with big air spaces between them.
Okay, thank you.
Spongy. Oh, what do we call the cells in the middle of the leaf? Mesophyll, middle of full leaf. Chlorophyll, chloro, green, full leaf. Okay. Right, so this is the mid-vein. So what's this all going around? It's different to the model. I think it's all sclerenchyma. Here they've got sclerenchyma top and bottom, and then parenchyma. Okay, what's at the top? What are these rays? That's the xylem, vessels and tracheas. Oh wait, no. Yes, that's right. Why did I was looking at this red. What's going on? Okay, yes, that's right. So xylem at the top. I was thinking, well, I've got this upside down. I don't. What's this here? Phloem below. There's phloem. And this layer? Lower epidermis. Guard cells. So this isn't green, obviously. Just the guard cells. You can see the stoma. So water moves up the plant, into the cells, through cells, through cells, through cells, transpires out. Oxygen, CO2 in, CO2, oxygen out. Okay, yes.
Ma'am, sorry, what's the cells around the vein part?
I think they're sclerenchyma because I can't see unevenly thickened walls like parenchyma. I can't see this. I see big sclerenchymas, but I wouldn't ask you stuff like that.
Oh, okay.
But I think these are sclerenchyma. In some veins, they only have sclerenchyma. In some leaves, there's only sclerenchyma around the vein.
Okay.
Depends. See the little vein here? There's sclerenchyma, xylem, and xylem and phloem. No branch vein. Okay, are we happy?
Yes.
All right. Then I gave you colouring in. Did you all do it? Where's your notes?
I threw it on now, but I have to remember.
Did you all colour in?
Yeah.
Alright, so let's have a look for that one. The line diagram. I mean, normally we do it here in class. Easy. And I've actually got this all on PowerPoint as well. Maybe I should go in first. Okay, let's do it here. Oh, okay, this is just not what I mean. Alright, so there's think of the root, a cross section through the root where the xylem and the phloem is. And then we just drew a few. So around the edge of the root, drew a few cells. There's epidermis with a little root hair. But obviously root hairs, they're only going to be near the end of the root, not everywhere. Okay? There's the epidermis. There might even be parenchyma below the root, if it's a nice big strong root. This is cortex of parenchyma, so lots of little air spaces between the cells. There's the two layers that you were talking about. Okay? So there's the endodermis, these cells. Modified parenchyma cells, you know, normally there's a cellulose cell wall all over here. Sorry, I'm getting hot. Oh. Okay, so normally there's cellulose all over, but these cells become modified and they develop this layer. It's almost like taking a big elastic band and putting it around here. Do you remember suberin, the fatty substance? And it doesn't like water. So if these cells are like this, all the way through the root, if they like, whoa, they like that. So now water goes in, across by osmosis from cell to cell to cell to cell, through the middle, and in fact, the cells here secrete salts here. So what follows salt? Water from this side, and then it goes into the xylem and up the root. Okay, it's almost like a mechanism to make sure that the water goes in here, the salts are secreted here, into the that tissue, and then into the xylem and up the root. And there below that, that is the pericycle. Lots of little dividing cells. All the way down, they can make branch roots. We'll have a look at the PowerPoint just now. Yes? Mm-hmm. Okay. Then you've got parenchyma filling up in between the xylem and the phloem. Xylem, what are these cells here? Big ones. Vessels, tracheas. Phloem, and I've drawn some vessels and sieve plate and companion cells. Okay, so that's the root. And the number of arms of the xylem could be different. Oh, there's the branch roots. I was showing how the meristematic cells can grow and make a big branch root to help stabilize the plant. All right, guys. Okay, dicot. Looks exactly what we've been doing. So, epidermis on the edge. There's epidermis, but now there's a waxy cuticle. There could be a layer of parenchyma below if it needs strengthening with the unevenly thickened walls. Then there's parenchyma with chloroplast. Further you go in, no chloroplast, maybe bigger parenchyma cells. There's a vascular bundle or vein, and then I've just coloured in here. So on the outside, those sclerenchyma fibres. All right, like that. Down the side, yes.
Ma'am, will a cucumber have a layer of parenchyma under the epidermis?
I think so, because it's quite strong. Yeah. Then you've got the phloem, the sieve tubes, the companion cells. There's a little bit of meristematic tissue, and that eventually will form a ring, and then it makes wood. Do you remember that story? Mm-hmm. Secondary thickening. And then there's the xylem. I've tried to draw xylem vessels and tracheas. And there's the pith. Okay. All right, guys, I know it's time. Right, the leaf. Okay, so the leaf, now we've got the whole cross section through the leaf. Alright, so I've drawn epidermis going around here, and I've shown guard cells with little openings, the stomata, there's quite a few stomata here. This layer, palisade parenchyma, spongy. Together they are mesophyll. And we've got upper epidermis. Oh, why did I cut it here? Upper epidermis, lower epidermis. This is, look, I haven't drawn collenchyma. I drew the one not from here. I drew, yeah, so it's sclerenchyma. I could draw some collenchyma there if it was there. Okay, and this is xylem. What are these though? Bigger ones? Vessels. Tracheates. Okay, this is? What are the big ones? Sieve tubes. What's that little dotty thing? Sieve plates. Okay, and the coloured in ones in between? Companion cells. There could be a thicker waxy cuticle at the bottom, and that part is the main vascular bundle, the mid-vein or the mid-rib, and these are the little branch veins. Yes.
At the top.
Yes. Sorry?
You said at the
Oh,
bottom.
yes, I did, didn't I? Yeah. Thicker at the top, thinner at the bottom. Yes, sorry. But a water lily leaf would have?
Thicker at the bottom.
Well, not even, because there's water there. It doesn't have to worry about transpiration.
Oh.
And at the top? Thinner, or yeah. Okay. Oh, and then here I drew, there's the guard cells, but in cross-section they look like little circles. See there? Orientation. Oh, and then I wanted to mention, and I think I did in the video, we say that leaves are dorsiventral. Okay, so which is my dorsal side? Remember, like a fish, they've got a dorsal fin, yeah? Ventral faces down. So in a dog, this is dorsal, ventral, but we stand upright. So we bipedal. So leaves have a distinct, we have dorsal ventral, they also have a distinct dorsiventral, dorsal and ventral orientation. The dorsal part, that's where the palisade parenchyma is. The ventral part, where the spongium, where there's guard cells and the phloem below. It's just a nice word, okay, dorsiventral. It's not in the notes anywhere. Okay, happy?
Yes, ma'am.
Alright, so now, this I need to print, I can do one. Happy. I can't remember what happened now. Okay, let's see what else we've got. Oh, I wanted to show you. I'm just thinking, where did I put it now? Plant tissue, so it's a PowerPoint I'm looking for. And I had it open just now and the electricity went off. Sorry guys, just give me a second. Oh. Tissue, there we go. Okay, we've got it. Oh, it's PowerPoint. So I can use that one. Alright, so now okay, so that's what we started off with. Remember I said look at the plants. And it had different tissues, organs and systems, and you had to try and name them. So now we've really figured out where everything is. And that's, that's why I did the line diagrams with you, because we need to figure out where everything is. So it reminds me of a map, of like a botany map, what we've just shown. But that's really complicated. So the ones that I've just shown you, the line diagrams are a little bit more simplified, showing demarcations, areas of where the tissues are found. Not every single cell. And then you can, that's what we've just revised now. So the cross-section, let me roll this here. Okay, I need someone to, anyway. So there's cross-section through root, cross-section through stem, cross-section through part of a leaf. You can use this as revision. It's just a nice diagram that I found on the internet. And there we go. So right at the tip of a root, there's a root cap. There's the meristem, undifferentiated cells. They haven't yet become the tissues. Well, there would be epidermis, parenchyma. Further back, you get the xylem and the phloem and the root hairs. And then further back, you get those big branch roots. And there you can see the Casparian strip around those cells so that water can pass through. Okay? And there's a lovely one. Remember this one?
Yes.
Okay, so how water passes. And in your textbook, there's 3 ways, symplast, apoplast, and I can't remember the other one now. 3 ways that water passes through. People, we're not interested in that. It can go through the cytoplasm, symplast. It can go through the vacuoles, vacuola. It can go through the cell walls only. It depends on how lazy the water is. All right, but I'm not going to ask you that. The water moves by osmosis from the soil to the root head, high water concentration. To next cell becomes high water concentration. To next cell, remember that? Right, across, through the cells with that Casparian strip around, that suberin fatty layer, through the middle of them, through the pericycle, parenchyma if there is, and into the xylem, and then up. And then we use this little, remember this? About how little narrow cells naturally allow water to go up them. Okay, we'll look at that next week. Do you not remember that? Capillarity. Oh my word. Alright, I did show you that. And there in the middle there, you can see the xylem in the middle. Okay, yes.
I don't really understand what the function of the Casparian strip is.
Yeah, let's just think now. I'm just trying to think if it's in the other part one about support and transport. Okay, let me do it now then we are clear about it. So it's these cells with the suberin layer, like a thick elastic band going around. So instead of there just being cellulose, there's suberin, a fatty substance on the side walls. And these cells are arranged like that. So can you see now that water has to go through the middle, and because the cells secrete salt, water will go definitely through the middle, into here and into the xylem and up. It kind of prevents the water from moving sideways and backwards. If there's salt here, the water goes through and there, it's not going to go back. You know what it reminds me of? Grade 9. Bicuspid, tricuspid valves, where they only allow the blood to flow, you know. You remember that?
Mm-hmm.
Okay. Don't you put your phone away? Alright, so that's the role. Directing the water into the middle where the vascular system is so that it goes into the xylem and up. Is that okay? Alright, that's all I'm going to say about that. And here, I need a laser beam. You can see these parenchyma cells here. Look at them. They've probably got sugars or something stored in them, stained up. And you can see a little bit. Sometimes the suberin has stained up. There you can see the phloem. It's not nicely, it's not a nice line, but I can recognize phloem, and there's some parenchyma in between, xylem, vessels, little tracheads there. I don't expect you to recognize everything, okay? Just be familiar with. There's some more, same thing. Okay, this is, I think, is a younger stem, because there's not much suberin there. A little bit there. Okay. And we're going the wrong way, and then we do the diagram, and we color it in, and then there's a monocot. A monocot root looks very different, a grass root. I just showed you, look, it's different. Not going in there, we're not learning that. And then we get to the stem. So that's a very schematic diagram of a cross-section through the stem, with this, the parenchyma, phloem, xylem, the pith. Very schematic. Okay, just go through this quickly, and there they are. Alright, there's the vascular bundle. Oh, look, there's collenchyma, epidermis, collenchyma, all the parenchyma here, sclerenchyma, phloem, meristematic. These little xylem cells that haven't even become thickened. Can you see that? Then lots of nice xylem cells at vessels and tracheates. Okay? And there, another one. Going from this, oh yeah, look at all the collenchyma. There's collenchyma. This looks like sclerenchyma. Oh, it is, yes. Sclerenchyma, phloem, xylem. Right, I promise I won't give you very difficult ones. And look at the big air spaces between the parenchyma, the little triangles. And there's a monocot, so a grass stem. Now it looks like those little clown's faces. I just showed you for interest. There's so much variation. Okay, so these are veins. All of these are the veins. Looks like a forehead and 2 eyes and a nose. There's the xylem, and there's the phloem, yeah, and the forehead. It's just differences. But in the video I said, please don't ruin it. I'm just showing you. It's mentioned in your textbook, don't ruin it. Alright, so there's the difference. Monocot stem, grass, rose stem, the model that I've been using. And I think, what time's the bell go? Should we do this quickly? Secondary thickening? Can we do it quickly?
Yes.
Okay, so in the young stem, alright, think of a little now, planting seedlings that you go and buy from the nursery. There's just going to be the vascular bundles and a little bit of meristematic tissue in the bundles. This is actually later. So when those seedlings start growing thicker, then that little bit of meristematic tissue there becomes a whole ring, because the parenchyma in between becomes meristematic tissue as well. So you end up with a ring of meristematic tissue, and I don't require you to know that it's called vascular cambium. And I don't need you to know all these names here. Alright, so that ring of meristematic tissue is going to divide by mitosis, and it's going to make a ring of xylem to the middle and a ring of phloem to the outside. Have they shown that? No. They have, but no, they haven't. Primary, secondary? I don't like it. Okay, so it's quite hard to find a nice diagram of this. Okay, so what we've got here, and really just simple. How does a little plant, how does a little seedling, if it becomes a tree in 100 years, how does it make these annual rings? So there are the separate veins when the little plant is still green and small. Then the meristematic tissue there in the veins, meristematic tissue develops in the parenchyma, and you get a ring of meristematic tissue. So now it's going to make a ring of xylem to the middle and a ring of phloem to the outside. And they've sort of not really. They've shown the ring of xylem, they've actually shown two here. But okay, here we can see. So there, wait, there's the ring of meristematic tissue. Please, I don't want detail here at all. Just see what I'm basically showing you. And you can see here it's already made. There was the original xylem, the next year xylem, the third year xylem, the fourth year xylem. It's 4 years old. And here would be all the layers of phloem which become so squashed. And it becomes the bark of the tree. Yes? The epidermis falls off. And there's cork and phloem here. That's the bark of the tree. Okay, so original xylem, next year, following year, following year. The annual rings of xylem. Okay, shame guys, you're tired. Okay, and there we go, lots of annual rings. So there's the first year of growth, and then 1, 2, 3, 4, 5, blah, 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 up until how many years it's been around. And a rainy season is when it's a very wide ring of xylem, because it's got lots of water in the ground to absorb, and very thin rings of xylem, annual rings, when there's very little water, like a drought. And that's how they can determine, go back in years, cut down the tree, then it can't grow, but they can go back in years to figure out how much water there was. Alright, and there, let's see if I've got another, oh yeah. Okay, so there are part of 3 rings of xylem. And here you've got all the cork with the phloem and the cork cells. You know, they used to make wine bottle corks out of cork. Now they make them out of plastic. But it used to be from here. And there's the phloem that gets made every year. And I like this one. Okay? Sure. So we've got the cork and the phloem over here. And there's the meristematic region, somewhere there. And year 1 would be here, let's pretend, year 1, year 2, year 3. Look how much water was present here. And then it makes lots of vessels. And if there's not too much water, it makes more tracheas. And could you see that on the video when I held this up and you could see the little pinpricks? I didn't make those, they were there. Do you want me to come show you again? No, thank you. Okay. Okay. And then we did the line diagram, we've just gone through that. And we've done the leaf, there, and finished. Okay. So Monday we'll just do a little bit on transport. We've actually done root transport.
Yeah.
So we'll just do up and down, xylem, phloem, finished. Okay, now make something fun.
Yay!
All right. Yeah, I want to now, all the basic stuff put into a revision folder, because to go back to each lesson, even I was struggling.
Thank you, ma'am.
Because there's so much stuff.
And that helps.
Yeah.
Just